Today we're going to talk about gene therapy. Before we dive in, why don't we take a look at first what is inside the cell. Every basic unit of life has a cell. There are many parts of a cell, but in this picture we can see the nucleus, which is also part of a cell. Inside the nucleus, there is chromosomes. Inside of chromosomes, there are DNA, and inside of every DNA, there is a gene. A gene is a special cut of DNA and is a sequence of nucleotides that are programmed for something. A gene contains information for a cell to read. In this picture here, we can see the little sticks or a ladder or whichever you may describe if you see it. These are the nucleotides. Nucleotides are in base in pairs: the adenine, thymine, guanine, and cytosine. DNA can also be made in the form of protein. Proteins can be found in various forms, from the smallest hair strand and to what you see in the mirror. Proteins also come in different lengths. The size will depend on how long that DNA may be. There should be around 300 letters. That letters corresponds to the nucleotides. In this particular picture, we have the protein actin. Actin is a protein found in your hair. It should be shaped horizontally because it corresponds to what our hair looks like, straight. This another picture is called the immunoglobulin. This type of protein is found in white blood cells, while in the red blood cells you can find the protein hemoglobin.
In the problem in gene, we can associate that with hereditary feature of the family, the mutation in the damage to chromosome, one of the family. For example, the brother has acquired a disease. First and foremost, you need to look at it with the hereditary connection. Say that only the mom and the grandfather have it, while everyone else does not. This explains that you also acquire the dominant gene for it. Now this is where gene therapy comes in. What is gene therapy? Gene therapy is a transplantation of normal gene into cell in place of missing or defective ones in disorder to correct genetic disorder. The main purpose of the gene therapy is to remove, replace, and repair any gene defect that anyone has, or in the internet term, gene therapy is a medical field which focuses on the genetic modification of cells to produce a therapeutic effect or the treatment of the disease by repairing or reconstructing defective genetic material.
While deletion is only one type in repairing and insertion of the new genes are also done in different methods. Vector is one of the 3 examples on how to insert a gene to a body. As you can see, we have the harvested cell from the subject that is claimed needed to be fixed. The cell's genes will be fixed, but in order for it to spread to the body, this will be delivered through virus on the bone marrow. That virus will infect the other cells with a new gene. So as the bone marrows continue to produce cells, the new gene is also spreading throughout. Second method is electroporation. Electroporation, by definition, is the physical transfection method that uses an electrical pulse to create temporary pores in cell membranes through which substances like nucleic acid can pass into cells. In this picture, we can see that we have this pregnant rodent. In order to insert new genes for the experiment, this has used the electroporation method to let DNA pass through the body and the cell.
Types of gene therapy. Plasmid DNA. Circular DNA molecules can be genetically engineered to carry therapeutic genes into human cells. Viral vectors. Viruses have a natural ability to deliver genetic material into cells, and therefore some gene therapy products are derived from viruses. Once viruses have been modified to remove their ability to cause infectious disease, these modified viruses can be used as vectors to carry therapeutic genes into human cells. Bacterial vectors. bacteria can be modified to prevent them from causing infectious disease and then used as vectors to carry therapeutic genes into human tissues. Human gene editing technology. The goals of gene editing are to disrupt harmful genes or to repair mutated genes. Patient-derived cellular gene therapy products. Cells are removed from the patient's genetically modified and then returned to the patient.
Gene therapy is an experimental strategy for treating and preventing disease by using genes. In the future, instead of requiring medications for surgery, doctors may be able to treat an illness by putting genes into a patient's cells. Although gene therapy is a promising treatment option for a variety of diseases, it is still a risky procedure that needs to be thoroughly researched to ensure that it is both safe and successful. Diseases with a genetic cause could be treated by supplying a correct version of faulty gene. However, in a practice, delivering new genetic material to a human cell is difficult. A promising method for delivery such as genes involve the use of DNA or lipid complexes. Yes, but only once we figured out how all of the genes interact. We only have a basic grasp of functions of a few genes at this time. We have no business messing with them outside of testing until we know enough to create an entire genetic sequence from the ground up.